Thank you all for coming. I'd like to introduce our panel. Aging also has the privilege of having reading glasses now. So our first panelist is going to be, you already know, Tom Insel, Director of the National Institute for Mental Health. Our next panelist is Dr. Marlene Behrmann. She's the George A. and Helen Dunham Cowan Professor of Cognitive Neuroscience, the co-director of the Center for the Neural Basis of Cognition, and a professor of psychology, and I should also say a very close friend and colleague of mine for many, many years. Our next panelist is one of our honored guests, Theo Arvanitis, a professor of e-health, innovation, and health, head of research at the Institute of Digital Healthcare at the University of Warwick. Our next panelist is Dr. Alison Barth, a professor in biological sciences and a member of the Center for Neural Basis of Cognition. Our next panelist is another one of our guests, Mengfeng Li. He's the vice president at Yat-sen University and the Chiang Kung Professor of Medicine and Dean of Yat-sen Shangshan School of Medicine. And our last panelist is Tom Mitchell, who is the E. Fredkin University professor and the head of the machine learning department. So I'd like to invite the panelists all to come up now, please. All ready? They don't know entirely what I'm going to ask them, of course. So, I'd like to start with something that leads off of what Tom Insel just told us about in the Brain Initiative. And in particular, Tom gave us some of the insight, and Subra did as well, into the potential impact for brain research in the coming decades and beyond. What I'd like to hear from each of you, and I'd like to go through all six of you, but maybe we'll lead with Tom Mitchell over on that end, is to share your thoughts on the kinds of advances, technological or otherwise. Tom emphasized technology, but also other kinds of theoretical advances and scientific advances that may occur in the next decade that are going to prove critical for improving our understanding of how the brain gives rise to intelligent behavior and then how we treat dysfunctions of the brain. So with that, Tom, could you start?
Well, let me start then with an observation. The study of the brain is fundamentally data-starved. Right? We're trying to study something with, your brain has 10 billion neurons, each connected to tens of thousands of others. And a typical day in the life of a brain scientist is maybe to collect data from some experiment, analyze that, and try to draw some conclusions and publish them. There's a key thrust of the new Brain Initiative, as we heard, was to fix that by collecting more data. And obviously, that's one direction we need to go. But the point I want to make is that there's a second half to the problem of getting from large data to a theory of how the brain works. And that is the problem of actually figuring out how we're going to bring together this very diverse set of experimental data into a model of the brain. So let me give you a quick example. With my colleague Marcel Just, over the last 10 years, we've been studying language processing in the brain. So suppose you're interested in understanding how the brain makes sense of language. We've been using fMRI and MEG brain imaging to do that. And along with other people worldwide studying this problem, there are already, in fact, thousands of different experimental data sets available that study one or another aspect of how the brain does language. Somebody will run an experiment where they present just single words to people and have people read those and record the brain data. Somebody else will run an experiment where they present a story and have people read that. Other people will run an experiment where they present sometimes words and other times just nonsense character strings that aren't really words. And they'll look at the difference between those two. So each of these scientists, in a kind of reductionist way, is trying to study one or another aspect of the phenomena of language processing. And we do have, good news, data analysis methods for looking at those individual experiments and making sense, figuring out, for example, what is in fact the difference in the neural activity if you look at a nonsense word versus a real word. But what we don't have, and this I think is a key opportunity, is data analysis methods, machine learning methods, modeling methods, that can simultaneously look at that very diverse collection of experimental data. The words versus nonsense words, the sentences, the words in foreign languages, the stories, they're all reflections of the same underlying neural circuits and neural phenomena. What we don't have today is data analysis algorithms that can simultaneously look at all that. That, I think, is a key thrust of, or one of the key thrusts that Dr. Suresh mentioned in his talk here at CMU. And so I just highlight that as a huge opportunity and a huge need.
So I'll just add before we go on that it certainly is one of the unique things about CMU that people like Tom are really becoming neuroscientists because of the fact that they're applying cutting-edge methods such as machine learning, and it's one of the great things about this place. And certainly in my own work, I do human neuroscience. We get frustrated because we think of this great experiment, and it turns out it would take 100 hours of someone laying in a scanner to do the experiment. We say we can never run that, so we need to figure out ways to actually leverage all the great work being done throughout the world. So Mengfeng, would you like to add?
Yeah, well, yeah, I, excuse me for my voice after this long travel from the other side of the earth. Glad to be back in town. I deeply share the perspective that Tom just brought us about how much and how badly new tools are needed to get this entire field to move forward. Let me use the example of Yat-sen University and something in China to hopefully to further that notion a bit. You know, China is a big country, and the country is, in terms of the brain research and in that area, it's quite unique in terms of the genetics, epigenetics, and features of the population. And it is unique in the today's industrialization and the environmental outcome for that. And it is unique in the culture after thousands of years history. And all those factors are contributing to the brain activities, contributing to the, of course, behavior, and also neurological diseases and other diseases. But they're not doing that in a simple way. To study them, we need a lot of new tools and technologies, and so that we can put what we're interested in together. For example, CMU University, as one of the leading universities in China, and our medical school as one of the best medical schools in China, we are seeing, well, the official number for our healthcare system of the university is that we're having 12,000 ward beds. We're seeing 12 million outpatient visits a year. And we're doing 170,000 hospitalized surgeries annually, meaning that we're cutting at least that number of human tissues off. And we're seeing a lot of neurological disease of patients. And for example, we have a very large data and tissue bank for autism. It's a 4 or 5 digit individuals were containing that big in our 3rd affiliate hospital, which has a big department for specialized in autism. And also on the other end, believe it or not, the fastest supercomputer in the world, now officially tested, well, it's called the THE2, is now on the campus of CMU University. Although we're not the best computer science school and university in the country. And using that, connecting these two part of things in on our campus has becoming a very challenging task for all the community members. And it is always hard for us to put multidisciplinary experts and researchers together domestically, particularly within one institution. That's going to be a long talk about this. But anyhow, what we are seeing in this BRAIN initiative is a great opportunity to bring the people who understand and can make tools and invent tools that can help solve the problems that we're facing in our medical and other areas.
Excellent. Alison?
So I was intrigued by Tom's Insel's thought that clarity represents this revolution in anatomy. And I would say I have really great confidence that in the next 20 years we will be able to assemble, at least for the vertebrate brain, and that might be for small vertebrates like fish, but it might even be for mice, maybe not quite for humans, that we'll be able to identify every cell type and every synapse type. I think we're going to get that data. And so I think the revolution for me is actually being able to identify the different types of cells and the different pathways and roles of activity in these different cell types for different behaviors and different diseases. And we're going to get there, but what it does is it puts biology at the forefront of the technologies that we can leverage to collect this data. I think we're past the point where we're going to collect enormous data sets and then try to statistically deconvolve them into function. I think biology will reveal for us very specific roles that are very stereotyped from animal to animal. And we'll be able to leverage what we know from very simple organisms then to the human brain.
Theo?
Yes, I liked your comment about the future, especially looking at the electrical and chemical circuits of the brain, because this is what we can see coming soon. And taking the idea of the development...
I'm not sure they can hear you. I think they want that microphone.
Maybe if I put my microphone a little bit higher.
Speak a bit louder.
Okay, can you hear me now? All right.
Even louder.
Louder? Okay. Can you hear me? Oh, I think my microphone...
It is on. It is on. I think they just want you to talk.
Okay, I'll speak louder. All right. Okay, so talking about chemical and electrical circuits, maybe we can learn from tools that we have used already for so many years through our functional imaging technologies. So, for example, I'll bring you what we do at the University of Warwick in collaboration with a lot of partners at the Midlands area, where we develop what we call in vivo biochemistry by using magnetic resonance technology, in particular spectroscopic imaging, to understand better metabolic behavior of disease. And we work in particular with childhood tumors in that initiative there, where we have learned to pick up interesting biomarkers from metabolism that tell us something about discriminating diagnostic cues for a complex disease like brain tumors in children, but also understanding the follow-up and the prognosis of this disease. Now take this at a different level. If we could transfer this particular type of information in understanding the healthy and aging brain, maybe we can have similar kind of biomarkers that tell us something about the normal functionality of the brain, or for other diseases like the important neurodegenerative diseases like dementia and the ones that are a burden to our healthcare system. So I think looking at the biochemistry and developing further our understanding of metabolism, but further molecular medicine through imaging technologies will be something that will be very important in the future.
Marlene?
I'd like to focus on three main points. Maybe I'll just hold this. What I take to be the three key ingredients that are going to be essential for our understanding of brain structure and function. So the first is the consideration that structure and function are gradually configured over the course of learning and experience. And so in order to begin to elucidate the dynamic changes that take place over the course of development, we're going to need to design technologies that will allow us to image the infant brain all the way through development. There are some prototypes on the market. We are going to need special surface coils. We're going to need special head coils, special technology that will allow us to image the infant brain with high resolution. These will need to be widely distributed, and we'll begin to aggregate massive databases that will allow us to examine how the brains change as babies grow up and learn over the course of development. There is another technology that I think is going to be absolutely crucial as well, and that will allow us, technologies that will allow us to image the brain under more naturalistic circumstances. So obviously behavior is much more than what can be measured in the bore of a 3 or 7 Tesla magnet. And so we need non-invasive mobile technologies. Again, there are some prototypes around for tabletop technologies that will allow us to image the brain under the full range of complexity of human behavior. There are 2 other key ingredients, which I'll just touch on briefly. CMU has got a long and revered tradition in both of these. The first is the interdisciplinary collaboration. We've got to be able to take data all the way from genomics, proteomics, through neural circuit, up to complex behavior, such as algebra and language. And in order to track through that entire pathway, we need to bring together disciplines such as computer science, molecular, people who are working at the molecular level, as well as people who are working on characterizing complex behaviors. CMU can and does utilize this kind of eclectic multidisciplinary approach, and it's going to be absolutely crucial as we move forward. The last point, and again, this is a plug for what CMU can do really well, does do well and can do well, is to be at the forefront of theory building. So science is not solely a bottom-up analytic approach. There are also conceptual and top-down motivations that determine the nature of the data that need to be collected. CMU can build theories. We have been doing so. One can use computational models to inform in dynamic real time the kinds of processes that at least humans undergo in the service of various behaviors. We can use these kinds of computational models as scaffolds and skeletons to improve theory building. And the combination of the tools, the collaborations, and the theories, I believe, is what's going to take us into the next phase of this major initiative.
Thank you. Tom, do you want to add some things now in addition to your talk? Or?
I have nothing useful to add. This
Oh, good. Excellent.
I think those are great ideas, and Marina,
So-
we're getting some of the granularity that this kind of a project needs, so it's good to hear.
So then I'm going to throw down the gauntlet to you and ask you to answer the first question. So you raised the issue of these brain mapping efforts for all the way from mouse up to human, and particularly the Human Connectome project and the European Human Brain Mapping project. And there's been, as many people in the audience and I know our panelists are aware, a lot of public critiques of some of these mapping efforts, but the question of how valuable they'll be. And I wanted to hear your thoughts and then some of the panelists' thoughts about how useful is it to map a system? I remember a quote many years ago, actually, from evolutionary biology, where an ill-understood model of an ill-understood world doesn't really get you very far. And so the question is, how will this be useful, and how do we leverage it into the kinds of theory that Marlene's talking about?
Yeah, it's really a terrific question. The one of my colleagues, Eve Martyr, who's done a lot of the most elegant mapping of a single circuit and a simple system, has said that at the moment when she had, to go back to Alison's point, mapped every cell and every synapse and every connection, she still couldn't predict the behavior of her system. That mapping can only get you so far. It may be necessary, it's not sufficient. So I agree with that. On the other hand, to take a really simple system like the human genome, which we spent a lot of time and energy mapping, I think what we learned from that were two things. One was at the end of the Human Genome Project, we realized that the information was going to come from variation, not from the reference sequence that was published in 2003. That was really wonderful if you wanted to know how humans were different from mice or flies. Not so helpful if you wanted to understand what any of these 3 billion bases actually did. And for that, you needed to look at the 3 million points of variation and how they played out, and you had to learn a lot more about the actual structure. So mapping didn't get you very far. On the other hand, that process and that project had so many spin-offs. And again, a lot of that was around technology development. And so it was through that project of mapping that we developed the sequencing technologies that have, I think, really been the basis for almost all of the major discoveries in biology over the last 5 to 8 years. So there's something to be said for embarking on these big mapping projects, even if the reference map you get at the end is almost the proxy. It's almost just a way to get to lots of other things that you can't predict.
Okay. I should note that as brain scientists, we think of the Human Genome Project as a simple project.
It is, absolutely. It's a linear project, and we knew what the end game was. We're going after something that is at least, who knows, 2-3 orders of magnitude more complicated.
At least. Alison, did you have something you want to say?
Yeah, I would say I agree with that in general. The map is going to reveal information that's good and solid information to riff upon, to build upon. And without that, it's very difficult to sort of constrain ourselves. You may not know how a horse walks by seeing that it has 4 legs, but you might be able to say it doesn't float. And I think we need these constraints. So one of the, having a thrust where you're really focused on something, for example, like the microcircuitry of a very defined system, like a cortical column in the brain, will give you some constraints to how that may function. It's not the end of the answer, but it's the beginning of the answer.
Tom, did you have something you wanted to add?
Yeah, I just wanted to add that if you look at this question from the perspective, if you assume that we would agree we want to have the schematic and we want to have a behavioral characterization of what's going on, and we want the mapping between the two. In the end, I think that's what we're after. If you think that way, then it's interesting to ask, what are the areas where there has been very significant progress in the last 10 years in terms of computational models of cognitive function? And I think 2 areas are where there really has been quite a bit of progress is computer vision. We now have very competent computer vision systems, for example, in your phone. That's why they find the faces and put a little box around it.
CMU company.
CMU company. And also language processing. There is a tremendous amount of resources going into computer understanding of natural language and a lot of progress there. Fortunately, those are 2 of the key questions about the brain, too. And so we already see work, for example, Mike probably is the biggest expert on the stage in this, but we do already see the beginnings of computational theories of human visual processing along with mappings to certain parts of visual cortex. And so looking at the big picture, it's kind of interesting to also take into account what are the areas where there is significant progress in computational models.
Anyone else have anything they'd like to add?
Yeah, maybe I could just...
Marlene?
If I could just build on that just a little while longer. So the idea of adopting theories and concepts from other domains, such as artificial intelligence as one example, is really crucial. There are other kinds of concepts from mathematics, from statistics, from physics that we are beginning to utilize, to exploit in the service of understanding the human brain mapping. A good example, and Theo and I have already discussed this, is things like graph theory. Of course, decoding and classification techniques are really useful to us at this particular point in time, but even more complex ideas from, say, percolation theory will really allow us to leverage in a non-linear fashion the data that we acquire from these high-resolution imaging studies.
Tom?
Mike, if I can jump in. When you, in the first round of questions, you asked the group kind of where they saw things going. And to go back to Alison's point, I thought it was really interesting. She started as a biologist by saying, we still don't have the basics. It is great to have the theoretical models, and we'll need that, and those can be important guides. But you want to get the parts list, right? And without putting words in your mouth, I think what I heard you say was there are just really fundamentals, like how many different kinds of cells are there. We know that in model systems like Drosophila or C. elegans, there's a tremendous amount of stereotypic anatomy. So you can go into a fly, the same place, same cell, one after another, and you know exactly what that cell's connected to, what it does. In the case of C. elegans, 304 neurons or whatever, every one of them has a name and identified. And those are animals that are able to do extraordinarily complicated behaviors, really, if you think about what they're able to produce. So the rules by which that happens are really interesting. The flip side of this is in trying to get the parts list for a human. We don't know if there are human-specific neurons. Do we have different parts than any other mammal or than any other species? And those are questions that really do beg for an answer, which we should try to pin down in some way. So I think there is the need for that kind of mapping and just getting the fundamentals done.
Cool. Anyone else like to add anything? Theo?
Maybe if I could add on that indeed the effort for mapping is kind of a bottom-up approach from the fundamentals to understand all the intricacies of the nervous system and the brain. But also we can learn by looking at top-down approaches. So by looking at existing diseases and look, for example, the effects of connectivity variations, let's say in Alzheimer's or epilepsy, by using surrogate measures that tell us something about the electrophysiology or the biochemistry. And through that, maybe we can push that information back to the fundamental to corroborate the evidence and make it stronger.
Terrific. Anything else? Okay, so as the next question, this is an aggregation of a question that was asked by many of the people in the audience, which has to do, we're going all the way from the lower level issues of technology up to now health. And there's a lot of interest, of course, in the health impact of all of this research, and Tom and Subra both alluded to that. And it's a very difficult question because, of course, we acknowledge that there's this incredible challenge facing the world because of the issues related to brain health. And there's a lot of things we can envision where we can say we can learn more about the brain and this will help solve some of these problems, but that this will help solve some of these problems is kind of a placeholder for, we don't know how to do that yet, but we're going to figure out how to do that. And it's obvious that possibly maybe at the circuit level or at the molecular level, maybe better, more focused drugs, but there may be many cases like traumatic brain injury or developmental disorders where there is no pharmaceutical intervention. So I want to hear your thoughts about how we as brain scientists and as basic researchers thinking about translation bring this into having real global impact that affects society directly. So, Marlene, you're nodding. Would you like to start?
It's a really great question. It's actually rather hard to answer. It's very provocative. I'll give you one example just from an endeavor that we've been involved in recently that may allow us to make a little bit of progress in this area. There has been a great sort of surge of enthusiasm, both nationally and internationally, to aggregate massive data sets of brain imaging data from different diseases. So, for example, we've just finished analyzing data sets from 1,000 different individuals of kids who have autism. So this is from 20 different sites internationally, have all archived their data on a server, and you can request permission to go and download these data. There are similar, Yat-sen has got similar kinds of massive data. The Simons Foundation has got massive kinds of data, especially genetic data that accompanies the imaging data. This idea of pooling resources on a global level, I think, is going to give us unprecedented insights into the brain, and will begin to give us a foot in the door to understand not only the mapping, but also the alterations and the deviations that one can see, at least in autism and, say, attentional deficit disorder, in the early stages of development. As Tom pointed out in his really important slide, these are the kinds of disorders that one sees very early on, but in fact, of course, have lifelong impact on the individual, on the family, and on society.
Tom wants to chime in.
So at the risk of being a little bit provocative, for those of you who aren't in the translational realm and who are thinking about getting into it, the important caveat is don't believe anything you're told by those who are in it. The diagnostic system that, at least for psychiatry, that people use is just fundamentally flawed. It's not based on any science or biology. It's based on a consensus. It's a political document, not a scientific one. And the therapeutics, which have largely been based on the development of pharmaceuticals, starts with a black box model of the brain and assumes that depression means you're low in serotonin and schizophrenia means you have a little bit too much dopamine, and it has absolutely no information about the brain as and the way that a neuroscientist would think about it. So you got to throw all that out and you'd start from the get-go. You'd do just as Marlene said, you'd start with, let's see where the data takes us. You'd go to someone like Tom and say, can you help us with machine learning to actually figure out what are the disorders? How many, we call this autism, and it's probably 10 different illnesses. Sure. So what are the right categories within the autisms that we should be thinking about? The way we think about cancer today, we don't talk about breast cancer. We recognize there are 6 or 8 different kinds that require different treatments. They have a different biology. And the therapeutics that we should be thinking about is very different. So if these are really not chemical imbalance problems, these are problems of neural circuits. So you can think of depression as a kind of arrhythmia, if you want, and schizophrenia as a problem around connections, and certainly autism as a connectopathy of some sort. Then your therapeutics needs to be, how do we tune circuits? How do we use the brain's innate plasticity to actually change the patterns by which areas connect, by which areas can synergize? And that's a really interesting engineering problem. It's a problem that needs the kind of insights that people at CMU would have for other systems. And unfortunately, that has not permeated at all translational space. So it does require a completely new view of the problem and a completely new approach. And actually, I think it's a fixable problem. We just haven't brought the right kinds of tools to it.
Theo?
So maybe if I could add on this train of thoughts, that we need to bring also the mathematicians and the statisticians to help us with pulling the data. I work in an area which is a rare disease, childhood brain tumors. If we collect data from our hospital, it's only a few tens of cases, which is fortunate for the children, but you cannot do proper analysis of anything you collect. So you need to pull data from across the board. So you need to cooperate internationally. We have a national network in the UK. We cooperate internationally. So for pulling all the data, we need the statisticians to help us with that meta-analysis of the information, to help us with standardizing the way we collect and we store that data and we prepare it for analysis. And as an example, one of the alumni of Carnegie Mellon is working at Warwick, Tom Nichols, and he has been working in the statistics department. And his effort is to develop a set of tools for supporting meta-analysis of functional MRI data for neuroscience projects. And that tries to avoid the problems of pulling data that is incompatible and that creates biases in the final results. So I think that is very important, and I can see immediately where that kind of initiative brings us together to solve these kind of problems.
Anyone else? Mengfeng, did you have something?
Yeah, well, I was going to add a bit. There probably another way of, or another dimension of thinking how this tool-driven approach could help the concept-driven science to get some real breakthroughs. I'd like to echo Tom's earlier point on that in his talk. We have a lot of, say, clinical specimens, and we have a lot of clinical information, a large volume of patients, and we can definitely. Well, we sometimes, a lot of times, we don't know what to look at and what kind of data we want to get from those. You can get genetics or genome sequencing, proteomics, and maybe a lot of other things. But I think if we understand what kind of tools are ahead of us and what those tools can lead us to, then we can start thinking better or more efficiently about what really we should look at from our what we have in the clinic and in biology. So this is, I think it is important, speaking of how this brain initiative and research can impact on what we're going to have.
Thank you. Anyone else? Alison?
You know, it raises this interesting question about being data starved, because I think that one of the things that has held neuroscience back is actually that it's so difficult to collect the data that you really are focused on a very specific hypothesis at all times. And once we break this barrier of not having enough data, it'll enable us to collect and analyze data in an unbiased way, not in this one part of the brain or for this one cell, but across the entire spectrum of brain function and also behavior. And I think then one of the big challenges will be developing statistical methods and being able to extract meaningful parts of that data to link it then back to disease phenotype or a treatment path. And that's very difficult, right? Because if you can analyze every different part of the brain or every different behavioral phenotype, you can't just do a series of t-tests to extract meaning. You need an enormous data sample. You need many, many individuals. And I agree that sort of collaboration then becomes, especially for human data, then will become necessary.
Thanks. So I want to follow up on something Tom said. He used the word plasticity. It seems to me that one of the other avenues, which was alluded to a little bit, we can think about a little bit more, is taking the brain's inherent plasticity and intervening in various ways for a whole range of brain disorders by allowing basically computational interventions, brain training. People have heard about things like luminosity, for better or for worse, tablet-based interventions that can be used in the home, in the schools, cognitive tutors, a large number of things, video games even. And I wanted to hear a little bit more about your thoughts about different levels of plasticity in the brain and the brain's ability to effectively, I hate the term heal itself, but at least improve itself either in all of us as we age or improve itself in ways when there's dysfunction in various ways. So does anyone want to take that on?
Well, if I were going to, if someone asked me that question directly, I would ask Marlene.
Okay. Marlene, at the next grant panel, you can quote that in your grant when you put in that IMH.
So learning obviously is absolutely key to, learning is just key to the dynamic changes that one sees in the brain, not just from infancy, but through the entire lifespan of an individual. One acquires new information, and that acquisition gives rise to a particular kind of, albeit limited, plasticity of the human brain. There are much more extreme cases of plasticity. Kids who've had an entire hemisphere of the brain removed, and there appears to be some kind of dynamic reorganization. If we could somehow harness our understanding of what those changes are and what the mechanisms are that promote those changes, I think we may be able to capture that. We may be able to sort of use the kernel of that mechanism in these behavioral intervention techniques. It will be fine to develop tutors, and we ought to go and develop the most adaptive, most cutting-edge techniques that we can. You can have them on your iPhone. You can use them all the time, except when you're driving. And the brain changes in all of those cases. It's not quite satisfying because we don't actually know what changes. When we really understand the fundamental mechanism, we'll be able to develop better tools in the service of intervention. I don't think we should wait, but I think it's going to be absolutely crucial to truly understand the neurobiology of learning and memory in order to be able to optimize intervention and remediation.
So this has got to be the last answer, but I want to follow up on that with Tom Mitchell a little bit. So Tom, I know you have a collaboration now in the whole Department of Machine Learning with Yahoo in looking at mobile device interactions, and I think of this as also a wealth of a data set. So not only can you intervene and have people's behavior modified through training and things like this, but the kinds of data you're getting from that is a model of human behavior at a scale that we've never had before. And we can, of course, ramp that up because, for all we know, Apple is taking data on all of our behavior all of the time. And certainly we know Facebook is at this point. So do you have some thoughts about how we can leverage that kind of thing into really coming back to addressing a lot of different kinds of issues that are facing us in terms of brain health?
Well, it's really a remarkable amount of data that's accessible through your cell phone. It has many sensors, and there's already interesting research going on, behavioral research, to see whether it's possible to detect someone who's in depression or not based on behavioral data from phones. And so I think to connect that opportunity to this discussion, maybe there are two, broadly speaking, connections. One is really to get symptoms from this high volume, steady stream of data from the phone for clinical conditions, depression and so forth, and then use those to identify and tap in. Those are the people you want to come in and scan if you're in some kind of project. But the other is really not a clinical condition, but just learning. So Marlene was mentioning that you could do luminosity on your phone all the time. But those kinds of signals from the system you're practicing on can indicate that, in fact, you have learned X or you have learned Y. These tutoring systems know that. And so that gives you another kind of easy potential behavioral link back into the education part of this. And I think actually those are two very broad things that at CMU we should be looking at, not just the clinical thing, but also the educational component, which is a very direct link in with the Simon Initiative and other of the strategic initiatives here at CMU.
Terrific. Let's thank our panelists again.